All right, can you actually hear me on Zoom?
Yes.
Okay, thank you. All right, well, welcome to the beginning of our summer seminar lecture series. So it's a little soft. All right, go ahead and use the microphone. All right, is that better?
Excellent.
All right, thank you. All right, thank you everyone for joining, both online and in person. Today we're going to start our CNS lecture series. So there's a nanofabrication lecture series that's Tuesdays and Fridays, I believe. JD is in charge and we'll send out details of that very soon. I'm in charge of the imaging group's lecture series, and we're starting today. And then it'll be every Wednesday after this. This week, Wednesday was a holiday, so we're doing it on Thursday. It's a chance for the CNS staff to run through what their areas of expertise are and for you to meet and put faces to who you might want to ask questions of. So my name is Arthur McClelland. I've been a staff scientist at the Center for Nanoscale Systems since 2011. I got my PhD in applied physics from the University of Michigan in 2009. And my area of expertise is really optical spectroscopy. So I found out as an undergrad that I enjoy playing with lasers and zapping things with lasers. So that's what I do. I joke that my job is zapping things with lasers and taking pictures of rainbows, which is pretty close to the truth. And people bring me all sorts of interesting things. So I have about 400 researchers come through my lab every year. And so I've seen almost everything. Moon dust, Neanderthal coprolites, plants, animals, ion engines, fuel cells, solar cells, quantum computers, you name it, has come through. Today, I'm going to start by talking about two different optical spectroscopy techniques. And these are not all the optical spectroscopy techniques I do. It's just what I can feasibly fit into an hour. So if you have anything that involves shining a light on a sample and getting information back, feel free to reach out and chat with me. So, pointer. There we go. So we're going to have a quick review about light and the nature of light. So half the time I'll talk about light as a wave, where it has an electric field and a magnetic field that are perpendicular to each other and mutually perpendicular to the direction of propagation. Half the time I'll talk about light as a particle. We call a particle of light a photon. Physicists denote it with this h nu, which is the equation for the energy of the photon. So that's our shorthand in physics for a particle of light, a photon. And then we'll talk a little bit about the guts of some of the spectroscopic techniques we do. And then if I have some time, we'll talk about some advanced data analysis, because in this day and age with computers helping us, we can generate enormous amounts of data very quickly, and then trying to figure out how to pull out the information we care about is the new challenge, rather than just collecting the data. So what is light in the wave picture? In the wave picture, light is this electromagnetic wave with the electric field perpendicular to the magnetic field. Maxwell, it's James Clerk Maxwell. The Zoom is hiding away. The name wrote down the famous Maxwell equations that tell us how the electromagnetic radiation propagates. And when we talk about light as a wave, we're usually talking about the wavelength of the light. That's the distance between two peaks or two valleys. And that's if we plot it in distance, or we can talk about the frequency of light, which is the same wave, just plotted against time. We can talk about the amplitude of the light, of the electric field of the light, and that's the power, the intensity of the light is the amplitude squared. So how do we talk about light acting as a wave? We see interference effects. So we can have constructive interference and destructive interference. So if we have constructive interference, two waves are in sync, so the peaks line up, and then the resulting wave is the sum of the two input waves. We can also have destructive interference, where the two waves are out of phase, so the trough of one wave is at the peak of the other wave, and then that results in complete destructive interference, and we lose all the energy. So light acts like a wave because it bends when it encounters corners. And this was described quite a while ago by Huygens in the 1600s and Thomas Young in the 1800s. And so there's a famous Young's slit experiment, which builds on the Huygens principle. If we have a pinhole and we send light through, we get these interference fringes. So we get a lot of light right at the center, and then we get destructive interference, constructive interference, destructive interference, constructive interference, because the light is bending at this corner and spreading out. And then as it takes different path lengths between going straight and going at an angle, it ends up interfering constructively and destructively. If light only acted as a particle, we would have just light in the middle, because the particles would go and they would either hit the barrier or go through the hole. And so we just get one bright spot if light only acted as particles. And we can set up these interference fringes. This is Young's double slit experiment. This particular picture is done with water. And so even with water waves, you can see these constructive and destructive interferences. So you can see the peaks and troughs. And when they hit this, when they, we're getting feedback. When they hit the double slit, they start bending and making spherical waves. And then at certain angles, they will destructively interfere. And so that's these flat lines in the water radiating out. This, of course, wouldn't happen if we had just particles going through. We wouldn't have these radiating waves. So waves really do interfere, and we really do see that with light. One of the ways we use this when we do spectroscopy is with the diffraction grating. So a diffraction grating is a series of grooves, and it will bend the light at different angles, and the different wavelengths of light will bend at different angles. And so we get white light spread out like a rainbow. And this I use in most of my instruments. So light doesn't have a charge, so I can't bend it with a magnetic field. It doesn't have any mass. So I got to be able to sort out the wavelengths somehow. And I sort out the wavelengths by bending them with a diffraction grating and spreading out the light like a rainbow. And then I take a picture of the rainbow. And so the internal part of my spectrometers, we start with an entrance slit. It goes to an imaging mirror, which images the light that we are interested in onto a diffraction grating, which spreads out the light like a rainbow. And then we image these on sometimes to an exit slit and only let one color through at a time. Or sometimes we send it to an array detector, which is just a digital camera, like your cell phone has a digital camera. And then we take a picture of the rainbow. And we know how many purple photons, how many green photons, how many red photons we have. In the wave picture of light, we also have the polarization, which is the direction of the electric field. Because it has a specific direction, we can use that to probe directions of molecules. So if molecules have a certain orientation, they will react with light with specific polarizations in different ways. Jumping to the particle picture of light for a moment. A particle of light is called a photon, and each photon has a specific quantized amount of energy. So we can go back and forth between the wave picture and the particle picture with the energy of the light. So the energy of the light is h nu, and that's why we use the little shorthand of h nu. But this is also where h is Planck's constant, c is the speed of light, is the speed of light over the wavelength. So we can relate both the wavelength to the energy of the light, and each color has a certain amount of energy, and each color will have a unique energy. A blue photon has twice as much energy as a red photon. So how do we know that light sometimes acts like a particle? I just explained all the ways we know that it acts like a wave. Well, the photoelectric effect is the classic explanation of when it acts like a particle. So if we have a piece of metal and we put it in a vacuum and we shine light onto it, and then we tune the color of the light from red through purple, at some wavelength, we suddenly start getting electrons popping out of the metal. If we turn up the amplitude of the wave, so we increase the amplitude of the electric field, if we have more energy in the wave, if you go to the beach and there's big waves, the big waves have more energy than if there's just little waves, right? So if we throw in big waves of light, we get the same effect. We only get electrons popping out once we cross a certain wavelength, a certain threshold energy. So this effect doesn't work with a wave picture of light. In a wave picture of light, if we turned up the amplitude, we should be able to turn up the energy we're putting into the system such that we eventually get out electrons even with red light, but that never happens. So we need to go to the particle picture of light, where each purple photon contains a certain quanta of energy, and that quanta of energy is what is needed to pop the electron off the metal surface. And then if we increase the intensity of the light, we increase the number of photons we're throwing at the surface, which will increase the number of electrons that are popped out of the metal. And so this is actually what Einstein won the Nobel Prize for, not the theory of relativity, but the explanation of the photoelectric effect and how lights have to be understood as particles. And we use this effect for X-ray photoelectron spectroscopy, which is one of the analytical techniques at the Center for Nanoscale Systems. It will tell you the elemental composition of your sample, or in photomultiplier tubes, which is a type of detector that I use. Yes. Yeah. Panel.
Projecting and also drag it.
Yeah. I'm apparently failing at Zoom. Anyone else? Yeah. All right, I don't know how to turn that off. Yeah, so, but turning up the light intensity only produce more electrons, not changing the wavelengths. All right. If we look at the Bohr model of the atom, which is overly simplistic, but we'll leave that aside. It works for what we're going to talk about today. We have a positive nucleus surrounded by electrons orbiting. We send in a photon. The photon can excite the electron. The electron can relax back down, and it emits a photon of a longer wavelength. And this is fluorescence. So this is just a laser going through a fluorescent dye. I'm setting in blue light, and we're getting green light coming out. So there are many optical spectroscopy techniques that deal with electronic transitions. We're not going to focus on them today, but I find it useful to put it in context because people have a little more physical intuition of fluorescence. I like to introduce these Jablonski diagrams, although I tend to call them energy level diagrams. All due respect to Dr. Jablonski. I find energy level diagrams a better description. So if we have different energy levels and we send in a blue photon, it's excited, it relaxes back down, it emits a green photon. So in my original cartoon, I cheated a little bit. And I sent in a blue photon and I got back out a green photon. But physics says we need to do conservation of energy. So what happened to the energy difference between the blue photon and the green photon? So in the Jablonski diagram, I've added in these vibrational energy levels, and we lose a little bit of vibrational energy due to heat. You can think of it as heat. And so that loss of energy corresponds to the difference between the input energy of the blue photon and the output energy of the green photon. So let's talk more about those vibrational energy levels, because that's really what I wanted to focus on today. So vibrational energy levels talk about the connections between the atoms. So we don't often deal with isolated atoms. You can deal with isolated atoms. People are building quantum computers out of neutral atoms or ions. But that's not the world I usually live in. So I usually work with molecules and atoms connected together. So I was trying to draw a picture of water. So water, the chemical formula is H2O. The hydrogen I can draw, one proton, one electron. The oxygen, I'm already too lazy to draw all the electrons. So as a physicist, we're going to cheat and jump over to this balls and springs method, this balls and springs model. The balls and springs model lets physicists go, oh, I know how to deal with that. We'll call it a simple harmonic oscillator. So the simple harmonic oscillator deals with just a couple of fairly old equations. And this is the only slide of equations I have. But we start with Hooke's law, which just describes the force on a spring, F equals minus kx. And we add in Newton's second law, F equals ma. So now we have two equations describing force. Force on a spring is minus the spring constant times x, the displacement. And force is mass times acceleration. We can rewrite the acceleration as the second derivative of position with respect to time. Set them equal, put in the time-dependent solution, and we get omega, the frequency of oscillation, is proportional to the square root of k over mu, where mu is the reduced mass. But this is telling us when we have a molecule, the frequency at which the molecule is going to vibrate depends on k, which is these spring constants, which type of chemical bond you have, single bond, double bond, triple bond, how electronegative things are, how tightly they want to stick together, and the mass of the atoms. So different atoms have different masses. We have the whole periodic table laying them all out for us. All right, So how do these actually vibrate? So once we do the simple harmonic oscillator, we can have what are called the normal modes and calculate how things are going to vibrate. At this point, I make everyone in the audience stand up just to wake you up. And we're going to do the water dance. So your head is going to be oxygen and your hands are going to be hydrogen. Up, up, up. Come on. I keep threatening to put disco music to this. I mean, I never get around to it, but. All right. So water has three vibrational modes. We have the symmetric mode, where the hydrogens are going in and out in sync. This is at 3,200 wave numbers. We have the asymmetric mode, where the hydrogen are out of sync with each other. That's at 3,400 wave numbers. And we have the bending mode, which is at 1,650 wave numbers. The two hydrogens are staying at the same distance and just rotating around the oxygen. All right, so if we can measure the frequency of a molecule and it has those three vibrations, we know that is water. Thank you, you may sit down. But everyone's awake now and everyone will remember the three modes of water. So we can do lots of math and group theory and figure out which ones are going to be Raman active and which ones are going to be infrared active. To be infrared active, it needs a change in dipole moment. To be Raman active, it needs a change in polarizability. I'll explain what all that means in a moment. But if we go back to energy level diagrams and we can look at some of the different transitions we can have, we can do infrared absorbance where we just transition from a ground vibrational level to an excited vibrational level. We can have UV vis absorption where we transition from a ground electronic state to an excited electronic state. So these are both absorption measurements. We're counting how many photons we lose after the light interacts with the sample. We can have Raman scattering, where we transition up to a virtual level and then transition back down. And in Raman scattering, it's an inelastic light scattering process. So we're losing some energy. And we're losing the energy because we start in the ground vibrational state and we end up in the excited vibrational state. And so we send in a green photon, we get back out a yellow photon. If we keep careful track of the energy we put in and the energy we get back, we can determine what this energy level difference is. And then that will tell us what the vibrational mode is, and we can make assignments and chemical understanding of our sample. You can do resonance Raman, where you transition up to an electronic state and back down. You can do fluorescence. So the chemists call it fluorescence. The material scientists and physicists call it photoluminescence. It's the same thing. We put in photons. We get photons of a different, longer wavelength back out. We can do second harmonic generation, some frequency generation, coherent anti-Stokes Raman scattering, stimulated Raman scattering. There's a variety of nonlinear spectroscopy techniques, which I'll touch on, but I won't focus on enormously. So after this long debate about whether light was a particle or whether light was a wave for hundreds of years, de Broglie finally said, everything's a particle and a wave. I have a wavelength, electrons have a wavelength. And he got the Nobel Prize in 1929 for writing out the de Broglie wave equation, the de Broglie equation, which relates the energy to the wavelength. All right, so if everything's a particle and everything's a wave, what can we use that for? So electrons are particles, they have a charge, they have a mass, but they also have a wave function. And so we can do electron diffraction and actually use their much smaller wavelength. And we can also think of phonons, which is the particle picture of vibrational waves. So here's a cartoon of a molecule with a vibrational wave going through it. And if you just look at the compressed part, you can imagine that can be modeled as a particle traveling down it. So these phonons in the molecule or the crystal are what we're coupling into when we do FTIR or Raman. we're coupling light into these phonons, and then we're losing specific energies, and keeping track of that lets us probe exactly how the molecules put together. So basically, optical spectroscopy boils down to, we shine light on the sample and observe some change in the light and the properties of the light to gain information about the sample. And the information can actually be encoded in quite a number of ways. It can be encoded in the wavelength or the amplitude or the phase or the polarization or the timing of the light. But you guys do this all the time. You go to the grocery store, you look at the bananas. Some of the bananas are green, some of the bananas are yellow. You shine white light onto the bananas reflect back light of a certain wavelength. You have a wavelength sensitive detector in the back of your eye. So you can tell these ones are reflecting back more green light, those ones are reflecting back more yellow light. Use some external information about bananas. You know, the yellow bananas have undergone a chemical reaction so that the starches have been converted to sugars and they're sweet and ready to eat today. The green ones are still starchy and they'll be sweet later in the week. So then you decide whether you need bananas for lunch today or bananas for lunch later in the week. So that's really all I do, is I shine light on samples and get information back. So to do the infrared spectroscopy, the infrared is usually done in Fourier transform infrared spectroscopy. So the Fourier transform is a mathematical transform. Fourier was a French mathematician in the 1700s, I believe. But because infrared light spans, well, the mid-infrared range spans 3 microns to 20 microns, sort of a very wide wavelength range. It's hard to find a material that works well across such a large wavelength range. So the diffraction grating trick that we usually use doesn't work so well, so we have a different trick. We're going to go back to the wave picture of light. And we'll start with our light source. So that's a glow bar. It's a broadband infrared light source. It's a silicon carbide. It's a piece of silicon carbide that we run current through until it starts to heat up. In essence, it's a not very good Thomas Edison light bulb because it doesn't even glow into the visible. It just glows in the infrared. But it gives us all the infrared wavelengths. We send this broadband infrared light into a beam splitter. The beam splitter splits the light into 2 arms. One arm goes to a fixed mirror and then just comes back. The other arm goes to a scanning mirror and comes back, and then the 2 beams are recombined. So when the fixed mirror arm and the scanning mirror arm path lengths are exactly the same distance, then we get constructive interference because everything is in phase. If they're half a wavelength out of phase, then we get destructive interference. Because it's broadband, different wavelengths will come in and out of constructive or destructive interference at different positions. So we get this interferogram on our detector. So at the 0 burst, we get everything's in phase and we get a strong constructive interference. And then as the scanning mirror moves, things will get out of phase and in phase, out of phase. And it dwindles off going both directions because it's broadband and different wavelengths will come in and out of phase at different points. So this is an absorption technique. So we do a background measurement first. We measure how many photons of each color we have without the sample. And then we introduce the sample and see how many photons we lose of specific energies. So with UV-Vis or with FTIR or all your absorption techniques, you need to do a background measurement first and then introduce the sample. So there's a lot of unevenness in the instrument response function. There's a black body radiation curve from the silicon carbide glow bar. There's a quantum efficiency curve of the detector. There's a throughput on the beam splitter. All these things need to be accounted for. But so we take a background measurement, and then we take a measurement with our sample, we get this interferogram, which is light intensity versus time. The Fourier transform converts from time to frequency. And then we have specific frequencies of light that we've lost due to interactions with the sample. So if the sample was water, we would lose three specific frequencies. We'd lose 3,400 wave numbers, 3,200 wave numbers. That's the anti-symmetric mode, the symmetric mode. And then we'd lose a bunch of photons at 1,650 wave numbers, and that's the bending mode. So this is why the water dance, which woke you all up, is important, because that will tell you where the three peaks that you would have for water is. So once we convert to this frequency domain, we can start putting interpretations onto it and having understanding of the molecular composition of the sample. So FTIR can be done in a couple different ways. Traditionally, it was done in transmission. This was the easiest way to think of it to start with. You start with a bunch of light, you send it through a sample, you see a diminished amount of light. This funny icon here is supposed to be the profile of an eye. It's physicist shorthand for detector. It actually took me a couple of years as a physics undergrad student to figure out what that was supposed to be. I knew it was supposed to be a detector, but it's supposed to be a profile view of your eyeball with the eyelids. Anyway, so the light goes to detector, we see how much light is lost. The problem is you got to make your sample thin enough to be IR transparent, which is actually hard enough to be tedious. A typical plastic bag is too thick. That's too much material to go through. So then people would grind up their sample and mix it with potassium bromide. And then you had to play this game of trying to balance the concentration in your potassium bromide solid mixture. It was a pain. So most people have switched to this ATR mode. And I'm gonna quickly just pull that up so it's not hiding. All right, so I pulled up the picture because I can't get Zoom to behave. All right, so in ATR, we use a trick called total internal reflection. We have an infrared transparent crystal. This can be zinc selenide, it can be silicon, it can be germanium, it can be diamond. The two ATR crystals I have in lab are diamond and germanium. You send in the light at such an angle that it totally internally reflects off the interface between the crystal and your sample. And then the light doesn't actually penetrate into your sample. We have just an evanescent electric field that penetrates into the sample. And that evanescent electric field can still interact with your sample, but it means we don't have to do any sample prep. You can mount ATR crystals from the top or from the bottom, doesn't matter. We had one from the bottom. There's an amusing story of a bowling ball company that worried about the surface chemistry of their bowling balls. So it would slide down the lane just right. They didn't have to do any sample prep. They just put the bowling ball directly on the ATR crystal, take the measurement, and move on. And that was their quality control. So you can do no sample prep and just contact it with the ATR crystal. Yeah.
Does the type of crystal matter, or when does it matter?
So the type of crystal matters because the penetration depth of this evanescent electric field depends on the index of refraction of your material and the index of refraction of the ATR crystal and the index of refraction of your sample. And that will determine how far into the sample you're measuring. My rule of thumb is you're going to measure about 500 nanometers in for a polymer, but it can be a little bit more, a little bit less, depending on which FTIR crystal you're using and what the index of refraction of your material is. People used to say, oh, well, people in the infrared world says, oh, 500 nanometers is the surface because we're no longer going all the way through the sample. But XPS says, oh, no, 500 nanometers is huge. You're well into the bulk. XPS does 10 nanometers. And then the people who do OJ says, oh, 10 nanometers, you're still 100 atoms in. OJ is 1 nanometer. And then Austin, who does the low energy electron microscopy, is like 1 nanometer. That's still way too far into it. We're going to do just the top surface. So surface sensitive is sort of a flexible term, but for most practical applications that you're going to use the FTIR crystal for, it's not going to matter the material so much. Diamond has become popular because it's fairly robust and holds up well in a shared use lab. Germanium has a higher index refraction, so it penetrates less, and so it's a little more surface. But really, if you are interested in surface chemistry, there's other techniques we have that will get you much more surface specific. Sorry, let me pull up the other picture that's hiding. Yeah, where does my mouse want to hide? Fine. And then the other mode we can do is just reflection. So we can shine the light down onto the sample and look at the wavelengths that are reflected. If there's strong absorbance in the sample, then we won't see those frequencies in the reflected beam. So this can be very useful if you don't want to touch your sample, if contact is no good for your sample for various reasons. All right. There's my... Just as sort of one example of the things that comes through is Dr. Joel Sohn is interested in chitin. It's a biopolymer that's widely found throughout the animal kingdom, everything from fungi to crustaceans and insects and mollusks. A polymer is like these kids' toys. So if you have one unit, you call it a monomer, but you can click them together to make chains. That would be called a polymer. So there are two different crystal structures of chitin, the alpha form and the beta form. And in the squid, the sea creature, actually uses both forms of chitin. And you have different physical properties with the two different physical forms. So in alpha, you just have all the chains aligned the same way. In beta, you have this anti-parallel arrangement of the chains. And so this anti-parallel arrangement gives you a lot more flexibility in terms of the polymer. So the squid uses this alpha chitin for things where it wants a lot of strong structural mechanical properties. So in its beak, squids have a beak sort of like a parrot beak where they're biting things. It uses alpha chitin. But in the pen, which is its sort of backbone, it looks like a feather quill pen, which is where it's got its name. It needs that flexibility to be able to move its body. It doesn't want a really rigid skeleton, so it uses the beta chitin. So this led to sort of an investigation like, all right, in the beak, it uses alpha chitin. They have like this little raspy radula, which they grade things up with. That turns out to also be alpha chitin. And this whole digestive tract is all chitin, but it transitions from alpha chitin in the front to beta chitin in the stomach. And so people have been looking at squid for thousands of years, but trying to understand exactly how it makes the different biopolymers and how it uses them. And is it a genetic encoding? Is there genes that are turned on and off and expressed in certain body parts? Or is it mechanical? Is there some way when it expresses the chitin, it aligns the polymers, the polymer chains in different ways? Squid are also very weird because this esophagus goes straight through the center of their brain. Their brains are actually shaped like a donut. And so imagine trying to swallow going straight through the center of your brain, and then you don't want a lot of flexibility there because you don't want your brain to be squished as you eat. So using FTIR, I can take my micro ATR crystal and sort of walk along the esophagus, getting into the stomach. And I can take spectra at each point. The ATR crystal is about 125 microns across. And if you look here, you can see differences in these peaks, and those peaks correspond to differences in the bonding. Oops. So even though these chains are the same chemical groups, the same chains, but they're arranged differently, we're sensitive, the phonons that we're probing are sensitive to the arrangement, whether they're arranged in parallel or in anti-parallel. And so we found that instead of there being a really sharp alpha to beta, there's sort of this transition zone over 800 microns. And this is still an ongoing research project trying to understand how the squid controls the crystalline form of the chitin it uses. Another interesting project that came through was the library came and said, we're doing this analysis of some of the historic photographs in the Harvard Library collection. And this one has this coating that was just painted over the face, but the rest of it was left blank for reasons we don't know. Someone got bored and went to lunch and forgot to finish coating it. So they wanted to know what the coating was. But you can't take a sample, you can't touch it, it can't leave the library. So thinking about the different modes of FTIR, I couldn't just do transmission because the paper was too thick to get through the paper and the coating. If you try, you get these strong, they look like saturation, but really it's absorption. It means no light is getting through. And we couldn't take a sample and grind it up and mix it with potassium bromide. Doing ATR wasn't an option in this case because the ATR left these little dents on the paper. They're 125 microns across. They're about the width of your hair, but it was enough. It's visible to the naked human eye, so that was no good. So then we decided to try this reflection mode, shine the light down, get the reflected light back. Even though these are the same chemicals, the physics between the ATR technique and the specular reflection technique means we get very different looking spectra. And unfortunately, most of the spectral libraries people have are for this ATR mode. And so we had to generate our own spectral libraries we had to generate our own spectral libraries for this reflection mode. But once we generated the reflection libraries, we could go through the collection of the Harvard class albums, which is an interesting collection because every year Harvard put a class album into the library. And so it's a time capsule of what the current photography technology was at that year. And so then I dragged my FTIR microscope over to the Harvard archives, and we actually found a really good match between our modern gum arabic spectra in red and the historic photographs in the dotted blue. So for 160 years of it kicking around, it actually matched quite well. And so we could make positive chemical identifications without taking a sample, without touching the sample. And this has spawned into other projects looking at pigments and binders and things like that with the Harvard Art Museum. This is looking at Indian yellow, which is xanthanate pigment that was made from the urine of cows that were fed exclusively mango leaves. And then the British Empire forbid it in 1905, something like that. So there's a very definite cutoff on when it could possibly be Indian yellow. So it's used as a marker in artwork. All right. Transitioning on to Raman spectroscopy. So in Raman spectroscopy, instead of looking at the direct transition between one vibration level and the next vibration level, we sort of do this roundabout. We go up to a virtual state, which is just a state the molecule can't stay in. If you think of it as, all right, I have seats on the right side, I have seats on the left side, there's this aisle. You're not allowed to sit in the aisle. You can sit on the right, you can sit on the left. The molecule sees a friend, gets up, starts over, halfway across the aisle, decides to go back to its original seat. But the original seat is taken, so it takes a different seat. So that's sort of what we're doing. We're starting, we go halfway across the aisle, but then we come back to a different seat, and we're keeping track of the energy levels between them. So C.V. Raman discovered this effect in 1928 using sunlight in Bangalore, India. The laser was not invented till 1960. This is from his Nobel Prize talk. He was taking the spectrum of carbon tetrachloride, which I do with the undergrads in the physical chemistry class I help teach. It's hard in 2024 to take this spectra. It's not a trivial spectra to take, and that's even with modern lasers and detectors and whatnot. So doing it with sunlight and glass filters and having to do darkroom photography, it's a very impressive physics observation. So as I hinted at, X-ray photoelectron spectroscopy will give you elemental composition. And elemental composition is nice, but we really care about the arrangement. Not all arrangements of carbon are equivalent. So no matter how nicely I photograph a piece of graphite, my wife's not going to want a graphite necklace. She wants the diamond necklace. If you give my office mate, Greg, both of these and ask him to take the XPS measurements, he'll tell you they're both carbon. That's what he can tell you, they're carbon. But there's a difference between graphite and diamond. So how do we probe that? So I would do it probably with Raman spectroscopy. So we have a laser, it comes in, it goes off a dichroic filter. The lasers you use for Raman are very narrow band, so every photon has exactly the same amount of energy. They go at the Center for Nanoscale Systems. I have lots of small samples come in, so I run everything through a microscope. The light goes down, interacts with the sample. Some of the photons undergo this inelastic light scattering and change wavelength because they've lost a quanta of energy to a phonon. We collect all the light back, we send it to diffraction grating, spread out the light like a rainbow and take a picture of the rainbow. And then the picture of the rainbow, we just take a line scan across, and we have lots of photons at this wavelength, lots of photons at that wavelength. We plot it in Raman shift. We set the 0 as our excitation line, so it's always a shift from wherever our excitation line is. And then we can chemically tell what the sample is. So going back to the diamond and the graphite, the diamond, every carbon sits in exactly the same chemical environment. It's a very pure crystal. And so we get one very sharp line at 1333. The graphite, it comes in sheets, right? You have a pencil, you're writing with a pencil, you're pulling sheets of graphite off. So it has in-plane chemical bonds, it has out-of-plane chemical bonds. And so we get multiple peaks, and they're much broader for the graphite. We can take our laser and map back and forth across our sample, taking measurements at each position, and then we can make a chemical map. So again, I didn't have to take a sample. I didn't have to do anything but shine laser light onto the sample. And here I can see that these parts I've colored in red correspond to this big peak here, which is the silicon. And these parts that are in green are these smaller peaks here, and they correspond to an aluminum oxide. Metals don't have a Raman transition signal because metals don't have a change in polarizability. The electrons can just go wherever. So metal oxides have a good Raman signal. Metals don't. So these metal lines in the computer chip don't show up, and they're just black. We can also measure the orientation of things. So people want to measure the orientation of their material. So here someone has this tiny little flake of, in this case, it was black phosphorus. And they wanted to measure which direction the crystal was. So if you take the polarizer and you run it through the Raman microscope and you swing the electric field in different directions, you'll have different responses depending on the crystal orientations. So when you plot it on this polar plot, you get these 4 lobes, and then you can say, in this picture, I know what my crystal orientation is, and then you can do interesting things once you understand the crystal orientation. 2D materials are a fairly hot and common topic in my lab. So you have your chunk of graphite again. You have these layers that you can pull apart, and you pull them apart with just Scotch tape. But it's hard to tell optically where there's just one layer, where there's 2 layers. So with Raman, you tend to have these specific shifts, and the different materials have different specific signatures. In MOS2, you have this broadening of the distance between these 2 peaks from 1 layer up to 5 layers. And graphene, if I go back, in graphene, you have the ratio of the G and the 2D band. And this is a multi-layer piece of graphite, not a single layer of graphene. If it's a single layer of graphene, this peak at 2700 will be twice the peak at 1550. And then here's combining, this was a botany project that came through, which was interesting. A group wanted to know what the sugar concentration in a red oak leaf was across a 24-hour cycle. So plants make sugars by photosynthesis. What happens at night? There's no sun. They're not actively making sugar. Does the sugar concentration stay the same in the leaf, which would denote there's some sort of active process? The plant is pumping sugar up into the leaf to keep the concentration the same. Or is it just a passive diffusive process? So then you would expect the sugar concentration to drop during the night and then come back during the day. So we had samples of oak leaves that were collected across 24 hours. Some poor grad student had to go up in a forklift every hour on the hour and sample a leaf and then snap freeze it for 24 hours. And then they brought that into lab. And we wanted to know what the sugar concentration was. So we had to do everything frozen. So we had liquid nitrogen on a metal block and kept it all frozen while we did it. And then we did this chemical map of the sucrose levels in this petiole structure. And it worked out that there was about 1,000 millimolars of sucrose in the phloem, and it was constant across the 24 hours, which determined it was an active process, which apparently nobody knew before. So yay, science. And we can do Raman maps of pigments from the art museum. The art museum will take cross sections of paints and try to see the different layers that the artist used. This was a Gauguin painting that they have in the Harvard Art Museum, and behind it, when they x-rayed it, they saw a different image. It's supposed to be a landscape with a horse. This is supposed to be a horse, and then this is a beach. I have trouble saying it, but anyway, he apparently didn't like it and then repainted it with this different paint, this different Polynesian goddess, and they wanted to understand what the different paints were that he was using. So they will take cross sections of the paint, either with a scalpel or with a hypodermic needle, and then they brought the cross section and we did Raman maps and determined where the different paints were. So one downside of Raman is that it's fairly inefficient. We get about 1 in 10 to the 8th excitation photons actually making it into this Raman scattering process. So there's a couple tricks for making it more efficient. One is this surface enhanced Raman, where we basically have metal, either metal nanoparticles or a rough metal substrate. And having a sharp metal point will interact with the electric field of the light and enhance the local electric field. And this will give us a stronger probability of Raman process happening. So this is the surface enhanced Raman scattering technique, and it's really just a sample prep technique. The microscope is all the same. The Raman spectroscopy is all the same. It's just in the sample prep, you have this sharp metal, sharp conductive surface. And if a molecule is sitting at the peak of the sharp conductive surface, you get a local field enhancement of the electric field of the light. And then you get a much stronger Raman signal. This was discovered in the late 1970s by Richard Van Duyn. And lots of people have used it to either lower their limits of detection. For a long time, there was sort of a competition on who could get the biggest enhancement factor. And people argue if they got 10 to the 5th or 10 to the 6th or 10 to the 7th. Not into that game, but you do get a strong enhancement, which lets you detect a much lower concentration. In general, fluorescence is bad for Raman signal. So here is 2 different Raman spectrum. One is collected with a red laser, one is collected with a green laser. The green laser is coupling into the electronic transitions. This was a piece of Kevlar that someone brought through. Kevlar is the polymer that's used in a bulletproof vest. We can see that we have some resonance Raman enhancement, but the Raman is sitting on top of this broad fluorescence background. And if we just switch to a longer wavelength light, we can get the Raman transitions without that fluorescence. So here we still have the Raman we can see. Often if you have fluorescence in your sample, it will overwhelm your Raman signal. So the way to get around it is often just to switch the excitation laser. So green laser to red laser reduces the Raman. So at CNS, between our 3 Raman microscopes, we have 5 different laser lines: 325, 405, 532, 633, and 785. And one of those 5 lines will probably get you either above or below the fluorescence. The other way to deal with fluorescence is to do the anti-Stokes Raman scattering. So we do have a home-built multi-photon Raman system that can do either coherent anti-Stokes Raman scattering or stimulated Raman scattering. So briefly, if the regular Raman, we start in the ground state and we end up in the excited state. The issue is that we're still going from a higher energy photon to a lower energy photon. This is the same as the fluorescence. But what if we started in an excited vibrational state, went up, and then went back down to the ground vibrational state? Then we're moving from a green to a blue photon. Moving up in energy means we don't have any fluorescence. The problem is that very few of our molecules are starting in this excited state. So the anti-Stokes is usually weak to the point of almost being undetectable. We can use multiple photons to pump our system so that we artificially put a bunch of our molecules into the excited state and then do this anti-Stokes transition. And once we do that, we can get 3D chemical maps. So when I first took over the lab in 2011, I found this slice of mouse brain sitting in a drawer. You never know what you'll find in the lab drawers. And so I did a CARS map of the CH2 stretches, which are the lipid stretches in it, just for kicks. But we can still do this sort of thing. But coherent anti-Stokes Raman scattering suffers from some non-resonance background. And so most people have moved to this stimulated Raman scattering. So we take our pump beam and pump the photons up, and then we stimulate them to come back down to an excited state. But if we have a very low noise laser, we can just count either the number of photons in the pump beam we lost, which would be stimulated Raman loss spectroscopy, or the number of photons in the stimulating Stokes beam that we gained. So that's stimulated Raman gain. It's very nice. It has no non-resonant background. And you can do very nice imaging. The downside of either CARS or SRS is that now we've tuned into a specific chemical bond and we're not getting the full spectrum. So we're losing a lot of information that we would get with the spontaneous Raman. But you can do things like you can compare the stimulated Raman scattering imaging to your standard H&E, and you can see that you can reproduce your H&E. And so now you can imagine going into an operating room, and here's the bright field image that the surgeon would see under the microscope. And then you could give them augmented reality with the stimulated Raman scattering and say, here's the boundary between the tumor and the healthy tissue. This is where you should cut. And then very briefly, because I'm running out of time, we'll touch on Raman labels. So in biology, you often want to know where things are going. And so you often label them with fluorescent tags, either fluorescent proteins or fluorescent dyes. But how do you label something like sugar? So knowing where the sugar goes is important, but it's a very small molecule. So if you put in a fluorescent label or a fluorescent protein, it's going to be larger than the molecule you're trying to probe. And thus it's not going to behave in the natural way. If you put this alkene moiety, this C triple bond on it, the C triple bond doesn't occur a lot in nature, and its frequency is shifted from all the other carbon bonds. So remember, the frequency of vibration was proportional to K, which was the bond strength, over M, which was the mass. So if we change the bond strength, we can shift it up into this cell silent window, and it's a nice strong peak. And so now we can have this glucose, and we can put in this alkene moiety, and we've labeled it, and we can track where it goes. And it's different than all the other sugars that are running around in the cell. So then we can play with that other parameter, the mass, right? So we have different isotopes of atoms. And so we can have 2 carbon-12 atoms in a C triple bond. We can have 1 carbon-13, 1 carbon-12, or 2 carbon-13s. And now we have 3 different labels. Wayne at Columbia has made this into like 20 different labels with various chemistries. But I think it's interesting to think about when, all right, so we had 2 parameters we could tune the bond strength, or we can tune the mass, and then you can actually tune both of them and move these peaks around to get different information. All right, since I'm out of time, I will stop there and take any questions.
Yeah, thank you very much for your talk. I'm just interested in knowing, can surface enhanced Raman spectroscopy measure small changes in biomarkers? Like you can measure small change within 2 months' time, you can increase the specificity of the test.
Right. All right. So I'll repeat the question for the people on Zoom. So the question is, will the surface enhanced Raman detect small changes in a biomarker? So the surface enhanced Raman is going to lower our detection limit so we can measure a much lower concentration of the biomarker. The changes in the biomarker are still going to be in the spectra. Let me jump back to the chitin. So here, this is a very small change in the chemistry. It's the same chemical, it's the same polymer. It's just whether they're parallel or whether they're perpendicular. And the change in the bonding between that results in peaks that are obvious even to the human. The second part of my lecture was about how to use sort of machine learning to pull out peaks, changes that are not as obvious as this, and you can pull out very subtle differences. Actually, let me jump forward. But the surface enhanced part is really just for detection limits. But when you're looking for changes in the chemistry, that's going to be changes in the peak positions. So here is an example of looking at FTIR of the different oils. So oils have a long CH2 backbone, and the differences are usually in the head group. But we can see the oils have different colors. Sesame oil tastes different than olive oil tastes different than corn oil. You can taste the difference, you can smell the difference. So chemically, there's some differences. If you just take the FTIR spectra with the ATR, they all look pretty similar because you're seeing what's the most common functional group, which is those CH2 groups. If we do what's called mean centering, so we take the mean of the entire data set and subtract it from each individual spectrum, we can then throw away what's the same and keep what's different because we only care about what's different. So for the biomarkers, it's what has changed in this biomarker that may be diagnostic of a disease. So if there's some change in the chemistry, we can pull out that subtle change in the chemistry. And then we can plot it in this principal component space. And now the safflower oil is well separated out from all the other chemistries. The safflower oil and the peanut oil and the corn oil and the olive oil. This data set was put together for by someone else, but I like it. And they were trying to see how much adulteration of the olive oil with corn oil they could detect, because unscrupulous olive oil dealers were adding corn oil to dilute it because corn oil is much cheaper than olive oil. And they could detect down to like 5% corn oil, 95% olive oil. And that about there is where the economics of faking your olive oil wasn't worth it. So they were happy with that. But so the changes in the biomarkers are in the peak positions or the peak amplitudes or the ratio of the peak amplitudes. And the surface enhancement is just lowering your detection limit to get down to physiological relevant concentrations. Does that help? All right. There were some questions on Zoom. Pull those back up.
We did have some concentration to the possible the lowest concentration for biomarker based on the Raman principle.
Yeah, so
With the concentration.
yeah, so we've seen improvements of sort of normal in lab, like 2 or 3 orders of magnitude detection improvement for biomarkers.
Raman intensity versus concentrations.
Yeah, so we've seen the concentration intensity change. The Raman intensity will change with concentration, and so you can back out concentrations. Yeah. From Zoom, it's from Zoom, what accounts for very different Raman spectral shifts for different excitation wavelengths for some materials and not others? So that is usually if you're coupling into an electronic transition and you're getting a resonance enhancement at specific, you're getting specific modes that are in a resonance Raman state rather than an off resonance state. So if you have a sample that's fluorescent and you're exciting it with something that would correspond to that electronic transition, you can get this resonance Raman state, which will make certain peaks much stronger than other peaks. If I go back to my Kevlar, where did my Kevlar go? So the off resonance in red looks very similar to the on resonance in green, but if you look at it carefully, you can see there's some differences. In this case, it's not a huge difference, but you can get some materials where you get a strong on resonance enhancement of just some of the peaks and not other peaks. What was the other question? How do you get Raman mapping images from pigment samples? Yeah, let's go. So to get the Raman mapping images from the pigment samples, the Art Museum prepared and polished a cross-section of the Gauguin Poems Babar painting. And we do point-by-point mapping. So we take a spectrum, move the sample, take a spectrum, move the sample, and then we get a spectrum from each of these points, and then we can take the spectrum that we collected and correlate it to a spectral library of the different pigments. It may be a little easier to think about with just sort of the three components here. The metal doesn't have any signal. The silicon has this single peak that shows up very strongly, and then we color code that the intensity of this single peak that's has the red cursors around it in red, and make this color-coded map of this is where the silicon is. And then the smaller peak in green that has the green cursor around it, we color code the intensity of that peak in green. And so say this is where the aluminum oxide is. So that's the same process that we did with the pigments. We're just using this information about what the peaks correspond to map out where the pigments are. All right. Those were the two questions from Zoom. Anybody else? Yes.
I think I didn't quite catch that. What was the mechanism in Raman spectroscopy that gives the signal in the spectrum?
Yeah. So the mechanism is we're coupling into vibrational transitions. So we send in a green photon, and every once in a while it will inelastically scatter, which means it loses some energy. And it loses some energy to the phonons, to the vibrational motions. So we send in a green photon, it goes up to virtual state, and then the molecule can come back down and stop in an excited vibrational state. So some of this energy between the different vibrational states goes into vibrational motion, and the energy is carried away by those phonons. But it means that the photon we get back out is a different color. Oh, yeah. So by measuring the energy shift, so we send in the green light, we collect everything we can, we spread it out like a rainbow, and we get a bunch of photons that are this color, a bunch of photons that are that color. And instead of plotting it in nanometers and wavelength, we plot it in this energy Raman shift. We have a lot of photons that were shifted by this much, so that tells us there's this specific phonon, this specific vibrational motion in the sample. There's this other specific vibrational motion. This was a piece of silicon, so this was the silicon-silicon vibrational stretch at 520 wave numbers. So the mechanism for our signal is this exciting the molecule to an excited vibrational state, and the energy being carried away by the phonon and measuring that phonon energy levels. And the phonon energy levels correlate directly to the molecular composition. Does that help?
Just one
Yep.
one question of interest. When you measure the DNA or the RNA molecule using the surface enhanced Raman spectroscopy, will that be more sensitive or less sensitive than the PCR test? The polymerase chain reaction. The polymerase chain reaction, you could measure like 10 copies.
Right. So in PCR, in the polymerase chain reaction, you have this amplification of the number of copies. And the Raman, we don't have that amplification of the molecules of interest. So if you're starting with 10 copies, we've got to measure from 10 copies, that's it, which ends up being challenging. The polymerase chain reaction, you start with 10 copies and then you copy them and you have this amplification, which is how you can end up detecting it. So if you're just trying to detect minimal number of copies, PCR is going to be the way to go, but it's expensive. It requires a fair amount of lab equipment and technical know-how and reagents. We all went through trying to get PCR tests when we had COVID, and you had to take the nasal swab, send it to the lab, wait 36 hours. By then, you were either healthy or dead. The Raman can be much faster, assuming it's within the detection limit, and the detection limit is going to vary on the molecule. Some molecules are going to have a larger Raman scattering cross-section than others, but we can put it under the microscope, and 30 seconds later, we have an answer. And there's no extra reagents, which were in short supply during COVID. The amplification takes time. It takes some hours. So they're not quite directly comparable. And if you're just going for minimum number of copies, the PCR is going to win. But if you're going for speed and flexibility, then Raman's going to win. And then it's a case of trying to get the detection limit low enough to be physiologically relevant. And then that's where these surface enhanced Raman games come in.
Very sensitive.
Versus
If for liquid and the solid sample, right on the surface. I like how students ask a question. It's your turn. Raise your hand, talk loudly.
All right. So I'll call it there. And if there's any other questions, people can come up afterwards.